this video we will discuss about N-Cadherin cleavage impact MMP9 levels. So in order to test this hypothesis, what they did is they cultured the cells with PMA for 8 hours and take out that media that they termed as CM1 culture media, and then they use this culture media to treat another set of cells, and the condition media that they took out from those cells they call them as CM2 condition media. And similarly for control they used cells which are not treated with PMA, and the first set of cells are called C1 and the second set of cells are called C2. So they will check the impact of this condition media, and then they will see whether this N-Cadherin cleavage has any impact on the MMP9 levels or not. So when they check this impact of this condition media and versus culture media for both the cell lines, that is NPC039 and NPC076, they are able to see that when they used NPC039, in the case of condition media, the cell invasion is significantly higher compared to the case of cultured media, and similar results they have seen for NPC076. And when they check the impact of the high cell invasions on the N-Cadherin levels, they are able to see that when they use the condition media, there is a decrease in the full length N-Cadherin signals for both cell lines, that is NPC039 and NPC076. Afterwards, once they see that the high cell invasions might affect the N-Cadherin full length N-Cadherins full length N-Cadherin compared to the control case where they don't culture the where the condition media is not from the PMA cultured cells. Afterwards they wanted to compare whether this condition media also has an impact on this MMP9 or not, and this time they not only check the MMP9 levels on the condition media 1 but also in the condition media 2 and the cultured media 2 also. And what they are able to see that when they use the condition media 2 or the second set of condition media, they are able to see enhanced MMP9 expressions compared to the condition media 1 for both the cell lines, that is NPC039 and then for NPC076. And these things they have quantified on the right hand side of the panel C, where we can see that with the condition media 2 the MMP9 levels is significantly higher compared to the condition media 1 treated cells. Now once they establish that in the case of condition media the N-Cadherin level goes down and MMP9 level goes up, now they wanted to see whether these two things has a relation or not. So in order to check that, they prevent the further cleavage of this fragmented and further cleavage of the fragmented N-Cadherin using secretase inhibitors. So when they use the secretase inhibitors and the cells in the control cells or in the cultured media cells, they do not see MMP9 levels, and when they used it for this condition media 1 only, they are able to see a high MMP9 levels, and they also checked it using gelatin zymography. In that case also they are able to see high MMP9 levels. Afterwards, when they used the secretase inhibitors with condition media, there is a diminish signal for the MMP9 diminishes in both the Western blot and the gelatin zymography, and this things is true for both the NPC039 cell lines and NPC076 cell lines. So from that it seems like that by preventing the further cleavage of this fragmented N-Cadherin, there is a decrease in the MMP9 levels, thus indicating that this N-Cadherin cleavage has some kind of rule towards this MMP9 levels, and by preventing this N-Cadherin cleavage, one can also diminish this MMP9 levels. This is what comes out from the results of this penalty and also from the results for the other panels that are presented in this figure. So I hope you understood the results that are presented in this figure and how they come up to the point that N-Cadherin cleavage has an impact on the MMP9 levels. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.